I'm going to erase. And sorry about using the whiteboard, guys. I had the doc cam ready for like pictures I wanted to go over, but it isn't working, so I had to resort to this. But I'm going to post everything that I was going to put under the doc cam on the WordPress website. And if you don't know what the WordPress website is, at the end of the session I'll write it down so you can check it out. But basically it's just a website where us tutors post resources for you guys. And there's a lot of good stuff on there, so I highly recommend checking it out. Okay. So I'm going to skip to the neuromuscular junction because I have a feeling, and I have this list on that website too. So this list, I went through all your slides and like picked out main topics. So I'm skipping some of the stuff that's like just neuroscientists, because you guys can look that up on your own. Okay, neuromuscular junction. Again, I like drawing a picture. The neurotransmitter for this guy is acetylcholine. The receptors, well, first the target cell or the postsynaptic cell, this guy, is a skeletal muscle. And then these are the nicotinic receptors. So definitely know like which receptors are for what. We're going to do the cardiac muscle over here, so we'll compare, and that's the muscarinic receptors, if you guys remember. This is going to cause muscle contraction, so it's going to cause an EPSP. An EPSP is excitatory postsynaptic potential, so it's the effect that the neurotransmitter has on the postsynaptic cell. So this is going to cause depolarization, so that's another way you can think of an EPSP is a depolarization of the cell. So just to go over like synaptic transmission and like how we actually release acetylcholine, we're going to have an action potential travel along the axon membrane. Remember, you have to have calcium influx to release neurotransmitters, so calcium is dependent for exocytosis. So remember that, just like link in your head exocytosis to calcium. That's going to cause, again, exocytosis, release acetylcholine. 2, acetylcholine bind to one receptor because there's 2 alpha subunits. There's 5 subunits in total, and there's 2 alpha subunits, and 2 acetylcholine have to bind to that receptor. So 2 acetylcholine bind to receptor. And then we're going to cause an EPSP, and how you can cause an EPSP is by having positive ions come in, because we're trying to get the membrane positive. So if you have positive ions come in, like sodium and calcium, remember that picture I drew up here? Remember which ions you have high concentration outside the cell and inside the cell? The only high concentration of ions inside the cell is potassium. Everything else is outside the cell. So sodium and calcium are going to come in. We're not going to let chlorine come in because that's a negative ion. Chlorine is also outside the cell. And we're not going to let potassium leave, because if something positive leaves, you're going to become more negative. So that's just a question of like, how can you cause EPSPs and IPSPs? The only thing I can think of that I'm missing is toxins for the neuromuscular junctions. So an agonist means that you're going to have the same effect as acetylcholine on this. So this toxin would also cause depolarization of the skeletal muscle and cause muscle contraction. So that toxin is nicotine. And it's kind of similar, if you just remember nicotinic receptors and nicotine, that can help remind you. Your antagonist is curare. So curare causes muscle paralysis because it's causing the opposite effect. Because normally we're contracting muscles, so the opposite of contracting muscles is not being able to move muscles. Okay. Cardiac, because acetylcholine is an example of having the same neurotransmitters with two different receptors and two different effects. So there's the cardiac muscle. Again, we have acetylcholine. The same process is going to happen. Action potential along the membrane, calcium comes in, acetylcholine leaves. This is going to cause an IPSP, which is hyperpolarization. So we're going to slow down the heart rate. Toxins for this guy. Is muscarinic. Oh, I didn't say that. The receptors is the muscarinic receptors. So then the agonist toxin is muscarine, if you notice the kind of the trend going there. Antagonist is atropine. So those are some toxins you guys might just have to memorize. Hopefully the picture makes it a little bit easier to see. That's the way like my brain works when I like work with that. Can I erase the left side of the board? Okay. So talking about neurotransmitters, there's three classes. We have amines, amino acids, and peptides. Your amino acids are your three neurotransmitters that start with a G. So you have GABA, glycine, and glutamate. Your amines is every like neurotransmitter you heard about before taking this course, like dopamine, histamine, serotonin, acetylcholine, adrenaline. Anything that ends with an E is an amine. So I'll just write a couple up. Serotonin. Okay. Peptides is everything else. So if you see a neurotransmitter, it doesn't start with a G and it doesn't end with an E, then most likely it's a peptide. The only one I know off the top of my head is substance P. But we don't really talk about peptides too much. Out of the amino acids, glutamate is your excitatory because it has a plus sign. It's the only one with a T. So that's your EPSP. GABA is your main IPSP in the brain because it has a B in it. Glycine is your main IPSP in the spinal cord. So there's different ways we can package neurotransmitters. Amines and amino acids, since they're smaller, they are synthesized and packaged in the axon terminal. And then peptides are synthesized and packaged in the soma. And Chang has a picture of that in one of your slides, and it's like, it shows the soma and the axon terminal, and he has like steps for each one, and it's like synthesized in the soma, packaged in the Golgi apparatus, travels down this. So like, just remember those are for peptides. And it does say in one of those things, like little boxes, that it's for a peptide. Okay, I'm gonna skip the G protein coupled receptor because that might take too long to draw. Precursors. So to talk about some of the neurotransmitters, how you make them and how you break them. So the one that he has on his slides with enzymes are is for serotonin, so I would definitely pay attention to this one. So the way we make serotonin is by tryptophan. Then we go to 5-HTP, and then we go to 5-HT, which is also serotonin. The enzyme is hydroxylase and then decarboxylase. How we make epinephrine, norepinephrine, dopamine, they are all in the same precursor family. So that one is tyrosine to L-dopa, or you can just call it dopa, it doesn't matter. Dopamine to norepinephrine, which is just the British way of saying adrenaline, and then epinephrine. And then lastly, glutamate to GABA is by the enzyme GAD, which is like glutamic acid decarboxylase. But if you just remember the acronym, then you can figure it out on an exam. Okay. Can I erase this part? Okay. So some of the main divisions of our CNS is our cerebrum, our cerebellum, and our brainstem. Our cerebellum is for movement coordination. Our cerebrum is for thought processing, and then our brainstem is for like everything else, basically. The right side of your cerebrum controls the right side of your cerebrum controls the left side of your body, but the right side of your cerebellum controls the right side of your body. So it's like, so how can I say that? Cerebrum is contralateral, and your cerebellum is ipsilateral. Ipsilateral. Okay. The meninges. So now we're going to talk about cerebellum is ipsilateral. Movement coordination. Yeah. All right. So your outermost layer of your brain, the one that's closest to your skull, is your dura mater. The middle is your arachnoid. And then your innermost is your pia mater. What's special about the arachnoid is where you have your CSF, which is your cerebral spinal fluid, and that is basically the juice that your brain sits in. And then what secretes the CSF is your choroid plexus. So I would definitely know that one. So real quick with CNS development. So you have your forebrain, midbrain, and hindbrain. In your forebrain, you have your diencephalon and your telecephalon. Your telecephalon is your two, we call them the bunny ears, but it's your two cerebral hemispheres. And then this middle part is your diencephalon. Your diencephalon is composed of your hypothalamus and your thalamus. Your telecephalon is your two cerebral cortex or hemispheres. So far, so good? Okay. Your midbrain, and there's like fancy names for each of these. I think this is like procephalon. I know this one's like Rano something cephalon, so look up those names, but for the sake of time, we can just call them forebrain, midbrain, and hindbrain. So in your midbrain, you have your tectum and your tegmentum. Your tectum is composed of your superior colliculus and your inferior colliculus. Your superior colliculus is your eyes, and your inferior colliculus is your ears. And I always remember that as like superior is above your ears. So I'm just going to write superior colliculus, eyes, inferior, colliculus is your ears. Your tegmentum has like, it's mainly for movement. It has your substantia nigra and something else. I'm not sure, double check on that. But it's mainly for movement for your tegmentum. And then lastly, the hindbrain is your cerebellum, pons, and medulla. Okay, so now we have to just talk about auditory, somatic sensory, and then chapter 24. And we're doing pretty good on time. And then at the end, if you have any questions or if you want to, I'm going to post those, but if you wanted to take pictures or, again, I'll write down that website. We can save that till the end. Okay, so auditory, there's like some pictures. I think pictures are the best way to study for this class. So just draw them over and over again until like you can do it from memory. But real quick, we have loudness, pitch, and timbre or timbre. I don't know. I never knew the right way to say it. Loudness is your amplitude of the sound wave. Pitch is the frequency of the sound wave. And timbre is the complexity of the sound wave. So this one would look like this. This would look like high frequency, and then this would look like that. Okay. So the first thing about auditory is knowing your anatomy. So this is your ear. You have your outer ear, your middle ear, and your inner ear. For your outer ear, you have the outside, this part is called your pinna. Then you have your tympanic membrane, also called eardrum. These little circles are kind of your ossicles, which are the smallest bones in your body. So you have your malleus, incus, and stapes, and those are your ossicles. And then this snail-like structure is your cochlea, and then this is your eustachian tube. So this is the first part of auditory, is can you label this? Because a good question would be sound travels through and hits this, and you have to know the order of everything. So definitely make sure you can do this picture from memory. Now, since this cochlea is coiled, we can uncoil it. And it'll look something like that. It's kind of wider here and then kind of narrows out at the very end. The beginning part of the cochlea is called the base, and the end point is called the apex. And right here, there's a little window, a little membrane, and that's called your oval window. And then you have your basilar membrane. It doesn't really show it too well in this picture, but it's narrow at the base and wide at the apex. So, at the base, you're going to have high frequency because sound waves are going to come in, they're going to hit the tympanic membrane, they're going to vibrate these ossicles, and then you're going to have mechanical vibration, and then you're going to have fluid vibration. So you have fluid moving in here. So in the beginning, you're going to have the most high frequency, and then it's going to trickle out to the lowest frequency. So we have high frequency and low frequency. And this is called place coding. It's just by knowing where that frequency is high and low. So this is basically knowing this is place coding. There's also another window, and it's called the round window. And the round window helps relieve pressure. Okay. So now, if we cut this in half, the cochlea in half, and look at it from within, we will find that it has 3 hollow tubes. On the top, you have your scala vestibuli. And I always remember that as vestibuli is on the top because you wear a vest on the top. In the middle, you have your scala media. And then at the bottom, you have your scala tympani. In your scala media is where you have the organ of Corti, and that's where hearing takes place.
That's where what?
The hearing takes place. Mm-hmm. So if we zoom in on the organ of Corti, we have our hair cells. We have a ratio of 3 outer hair cells to every 1 inner hair cell. Even though we have more outer hair cells, the inner hair cells are actually more sensitive because they have more spiral ganglion. So this whole thing is called the hair cell, but this top part is called the stereocilia, which is like the hair part. That's the part that's actually going to be moving when the fluid moves. So what's going to happen is, well, let's label this real quick. We have our tectorial membrane on the top and our basilar membrane on the bottom. So when there's fluid moving in the cochlea, these 2 membranes are going to be moving back and forth, and they're going to cause these little stereocilia on the hair cells to like move back and forth. And then that movement is going to cause an action potential, and then this is going to cause us to hear, which is really cool. So if we were to zoom in now on the individual hair cell, if you notice, I don't draw it all equal length. There's a reason that they're slanted, is because if it moves in one direction, it depolarizes, and if it moves in another direction, it hyperpolarizes. So if it moves to the tallest hair stereocilia, it's going to depolarize. If it moves to the shortest stereocilia, it's going to hyperpolarize. So in the stereocilia, we have this fluid called the endolymph. And then in the bottom of the hair cell, we have perilymph. In the endolymph, you have high concentration of potassium, which I know is weird because we're used to high concentration of potassium inside the cell. And then outside the cell, you have high concentration, or in the perilymph, you have high concentration of sodium. These stereocilia are connected through tip links. So that way, if one moves, they all move. So they all like flow together. So there's a mechanically ion-gated channel on the stereocilia, and it's mechanically because it's the movement of the fluid causes an opening or closing. So mechanically gated. To cause an action potential, we're going to have influx of potassium, a little bit of calcium. I noticed he added that on his slides, into those the mechanically gated ion channel. That's going to cause a depolarization. And then you're going to have calcium come in, and then you'll have release of neurotransmitter onto your auditory nerve, which is also called your cranial nerve 8. So far, so good for auditory? Okay. I have the auditory pathway. I drew it all out nice and pretty and neat, and I was going to put it under the doc-cam, but since the doc-cam isn't working, I'll just post it online so that way you guys can have it. So I'm going to skip that. Okay, so we're going to switch to somatic sensory. Do I need to leave this up for a second, or do you guys got it? Got it? Okay. So how do you guys feel for your final? Do you feel ready? Almost? Yeah. You guys still have time because it's not this Monday, but next Monday, right? Yeah. So you got plenty of time. Okay. So your somatic sensory is basically for touch, pain, vibration. I'm blanking out. Somatic sensory. So one part of your somatic sensory is your mechanoreceptors. And these receptors are on your skin, and they're for touch and vibration. So you guys have probably seen a picture, but if this was a cutout of your skin, so the top, like if this was a hair, like a hair follicle or something. So this is like looking within the skin. You have 4 mechanoreceptors. You have 2 that are deep within the epidermis, and then you have 2 that are on top of the epidermis. So the 2 that are on top, they respond to more like lighter pressure. And then the 2 that are deeper within the epidermis, they respond to more deeper pressure. Because if I like lightly stroked my arm, I'm not causing enough pressure and vibration to reach them. Does that make sense? So the 2 that are on the bottom, the one looks like an onion, and that is called your Pacinian corpuscle. And I, on an exam, had to know what these look like, so that's why I'm taking the time to like draw them out. And it's good to get an idea of like which ones are deeper in the epidermis and which ones are more on top of the epidermis. And then you have one that kind of reminds me of like a football shaped, and that is your Ruffini ending. And then you have the 2 on the top. One kind of looks like Pacinian, but just a little bit smaller. And that is called your Meissner. Corpuscle, I think. And then you have one that looks a little bit more disc-like shaped. And that is your Merkel's disc. So again, these 2 respond to light pressure, and these 2 respond to deep pressure. So we can kind of organize these mechanoreceptors into this nice convenient box. And I remember when I took this exam, I had a different professor, but when I took this exam, I drew this box on my exam to help answer some questions. So it's really nice for like organizing material. So on the top we have receptive field size. And then over here we have adaptation. We have small receptive field size, large receptive field size. We have slow adaptation and fast adaptation. So if you can just label that, and then I'll teach you like a little way to help remember like which mechanoreceptors go in which box. But that's just the gist of the labeling. So to help you remember which one goes in this column for small mechanoreceptors is your two that start with an M, because M&Ms are small little candies. So you have two M's. And then you have to decide, well, which one is slow and which one's fast. Merkel to me just kind of sounds like an old man. So it's like he would be slow. So we're going to put Merkel on the bottom and Meissner on the top. So then we have to decide between Vicinian and Ruffini which one is slow and which one is fast. Ruffini kind of reminds me of Rafiki, that monkey from Lion King, and he's an old man, so he's very slow. So I always remember that as slow adaptation. And then Vicinian is fast adaptation. So what does slow adaptation and fast adaptation mean, and what does small receptive field size and large receptive field size mean? So if you guys remember homunculus, the weird shaped man, if you notice his hands, his lips, and his tongue were really large compared to the rest of the body, because those areas are most sensitive because we have the most dense receptors there. So that's why like kissing and holding hands feels really good, because we're firing a lot of receptors. So on our hands, especially since our like fingers are very sensitive, we have small receptive field size. So they're very sensitive. But something like on my lower back, which doesn't need to be as sensitive, I'm going to have larger receptive field size. So what does this mean? On his slides, he talks about the 2-point discrimination test, which basically means if I poke this person's hand with 2 points and I ask them to tell me, do you feel one thing or two things, then there's a good chance, because these are such small receptive field size, that I'm going to hit two different receptor density size, and they're going to tell me that they feel two. But if I did this on someone's lower back, and I had my two pencils next to each other, and I told them, do you feel one or two, they're going to tell me that they feel one, because they hit one receptive field size. So if you see something like on an exam and you see that they're on like separate, then know that they're feeling two sensations, and if they're on the same, even though there's two pencils or whatever, then it's only one sensation. So again, Meissner and Merkel, they have smaller receptive field size. Vicini and Ruffini have larger field size. So what does adaptation mean? So slow adaptation means, well, first, fast adaptation means like, you know when you put on your watch in the morning and you feel it and then you kind of forget about it throughout the day, and then maybe when I do this and I move my arm, then it's like, oh yeah, my watch is there. That's slow adaptation. So you feel it when you put your watch on in the morning, and then you kind of forget about it during the day. Maybe you like shake your arm and you remember it's still there, and then when you take it off at night.
Wait, is it slow or fast?
Oh, sorry, that's fast. Fast adaptation, yeah. Because you're adapting to it really quickly, so you forget about it. Slow adaptation would be you would feel it all day long. You wouldn't have a break. It would constantly be firing. Okay, so let me erase this side. What makes it slow?
What would an example of that be?
Maybe like a really tight pair of jeans that you just feel all throughout the day, constantly, yeah. Yeah. So we have some different types of primary afferent axons, and if you remember, afferent going back to VEMDOS means sensory. So there's different types of sensory axons we have. So just looking at this picture, which one, and this is myelin sheath, by the way, and then this is the axon, and then this one you can see doesn't have any myelin sheath on it. Which one do you think would be the fastest? The one on the very left, right? That is your A alpha, and it is the fastest of the axons. Then you have A beta, A delta. I don't know how to do, or it's a weird symbol. I think it's actually gamma. I'm not sure, but I'm just gonna write, I think it's delta, A delta, and then your C fiber. So your A alpha is the quickest, so the function for this is proprioception. And I'll explain in a second what proprioception means. Proprioception. Proprioception is knowing where your body is located at all times. So like if I close my eyes and move my arm up and down, just because I know where my arm is, that's proprioception. So we need that to be the quickest because we need to know where our body's at all times because that's how we're graceful and everything. Our A beta is for mechanoreceptors. So those are these guys for touch and vibration. That's very important, so they need to be relatively quick. They're very similar to A beta. They're just a little bit smaller diameter and a little bit less myelin. A delta and C fibers, they have the same function. They're both for pain and temperature. But the thing is it's different types of pain and temperature. So your A delta, because A delta is going to be faster than this one because this one doesn't even have any myelin, is going to be for sharp pain and intense temperature. Your C fiber is going to be for achy pain and non-intense temperature. And examples of that is sharp pain would be like, he also calls it in his notes first pain and second pain. So first pain would be like stepping on a nail, like it's very sharp pain, it hurts really instantly. But second pain or achy pain would be like going to the gym and then the next day you're a little sore. Like it's not like immediate sharp pain, it's just kind of like achy and it's there and it's slow. Intense temperature would be like if you put your hand on a hot stove. You feel it's very hot, you remove your hand quickly. C fibers would be for more like if you were to step into a warm bath. Like you don't instantly go, oh, warm. It's kind of like, oh, this is nice and warm. Like it's a little bit slower. So that's why I wrote non-intense temperature and intense temperature. So there's a couple pathways. These pathways I think I can actually draw because they probably won't take too long. These are our touch pathways. Can I erase this? Okay. So the important thing with pathways, I know we didn't get to go over it with auditory, is definitely know where it crosses over. So we'll talk about that in a second with the dorsal column and the spinal thalamic pathway. So dorsal column, it's also called the dorsal column medial lemniscal pathway, but I'm not going to write that all out. But the dorsal column pathway and the spinal thalamic pathway. So the easiest way I like drawing these, he has the picture of it like with the brain parts, but I think it's easier just to draw boxes. So this is the spinal cord. This is the medulla, thalamus, and the cerebral cortex, which I'll label in a second. So we got spinal cord, medulla, thalamus, and cerebral cortex. Okay. So for the, and for both of them, at the thalamus, they're going to synapse at the VPN. Don't get that mistaken with the MGN, yeah, which is in the auditory pathway, which I know we didn't go over, but it'll be on the website. Okay. So with the dorsal column, the functions is touch, vibration, the 2-point discrimination test, and the spinal thalamic is mainly pain and temperature with some touch. So let's say we're on the left side of the body. We have a synapse at the spinal cord, and we know that this is going to be our sensory neuron or axon. So if we were just to make this clear, like left and right, it's going to travel up, synapse at the medulla, cross over, go to the VPN, and then synapse at the primary, or you can just call it the S1, the primary somatic sensory cortex, or S1. So that's the dorsal column pathway. So important to know, crosses over at the medulla. So in other words, I can say ipsilateral until the medulla, and then contralateral. The spinal thalamic pathway. Synapses at the spinal cord, immediately crosses over, travels up, skips the medulla, synapses at the VPN, and then synapses at the cerebral cortex. So the way I remember that is spinal thalamic pathway has like spinal cord in it. It immediately crosses over at the spinal cord. So it's immediately contralateral. Does that make sense? I think the boxes help break it down to make it a little bit easier. Okay, and then after this we can move on to chapter 24. Does that sound good? Okay. Am I good to erase? All right. Okay, so chapter 24 is all about memory, and there's two different types of memory. There's non-declarative and declarative. Declarative memory is for like facts and events. It's kind of like us studying for our finals. We're just trying to get the facts and events down. It's easy to learn and easy to lose. Like you can memorize a little fact, but then it's not going to last long unless you like constantly are rehearsing it. So non-declarative is, well, also this is more explicit. This is more implicit. Non-declarative can be broken up into procedural, memory, motor, which is like muscle memory, and then emotional responses. So for procedural memory, that would be like learning to play an instrument, riding a bike. It's not like declarative. Declarative is easy to learn, easy to lose. Non-declarative is hard to learn, but hard to lose. Like, it takes you a while to learn how to ride a bike, but then once you learn how to ride the bike, you're good. You're not going to really forget that easily. Motor is for like, again, muscle memory. So a lot of people who play sports can relate to that. And then emotional responses is like remembering that you're afraid of snakes, so to like avoid them or whatever. So in each part of non-declarative and declarative, there's different parts of the brain that corresponds to those. So for procedural memory, it's located in the striatum. And I always remembered that because, like, again, procedural memory is like learning an instrument. So it's like strumming a guitar. It's in your striatum. So like a little way to memorize that. Movement is going to be in your cerebellum, because that's where movement coordination takes place. And then emotional responses is going to take place in your amygdala. Whenever you see amygdala, just always think like emotional, fear, anger, stuff like that. So, okay. Declarative takes place in your medial temporal lobe. And the way I always remembered that was like when you're taking an exam, you kind of go like, oh, what's that answer? And you kind of rub your temples. So it's like medial temporal lobe is for declarative. So definitely know where each are located. And that's going to come into play when we talk about case studies. Retrograde versus anterograde amnesia. Amnesia is the loss of memory. So retrograde, I think retro, like retrospect, going backwards, you lose your memory from your past. And anterograde is you can't form any new memories. So lost old memories, can't form new memories. So a graph to help explain this, or it might be on a test question, I don't know. Retrograde would be like you maybe remember your childhood and then an accident happened, so you forget that period, but then you can make new memories. So an example of this, like for the movie, would be like Bourne Identity with Matt Damon, where he like forgot that he was an assassin. He kind of remembered a little bit of his childhood, but then he forgot all about his like assassin work, but he was still able to form new memories. That's retrograde amnesia. Anterograde amnesia would be like the movie 50 First Dates, where she like can't form any new memories. So hers would look like this, and then it would just scoop down, and she can't form. This is memories, by the way. And then this is time. Okay. I'm going to erase this. Okay, so there was a couple experiments in this chapter. So the first one was Lashley. And what Lashley did was he had some mice, and they were going through a maze, and it was like, so there was a mice maze, and after a while they learned how to go through the maze quicker and quicker just by getting the food reward. And then what he would do is he would take out lesions, and he noticed that that would affect the performance of the mice and if they can get to the food quicker. The only thing wrong with his experiment was what he concluded was that the larger his lesions, the more mistakes the mice make, which is kind of like, well, duh. But at the same time, he didn't say that like it was a specific region, which we know now there's different regions of the brain that like affect areas. So all he said was just that size correlates with more mistakes. The more he cut out, the more mistakes they would make compared to like a smaller lesion for that. So that was Lashley's experiment. IT cortex. Anytime you see IT cortex, that's always used for facial recognition. And also stores memory. He stressed that a lot on his slides. And they had an experiment with the monkey and like recognizing different faces, and there was a graph with it and everything. And yeah, I would look over those slides. But that's IT cortex. HM was a case study. So basically this boy had a lot of seizures, he had epilepsy, and they took him to the doctors and they removed, I have this on my sheet, I don't know if I can remember. It was, this is a case study of a boy, and he went to the doctors, and because for most of the experiments and case studies, just know what they removed and what the effect was. So they removed, let me double check on my notes. This is why I had it pre-prepared. They removed medial temporal cortex. It's kind of a laundry list for this guy, the poor thing, he had a lot removed. His amygdala and anterior 2/3 of hippocampus. So basically with HM, he had severe anterograde amnesia. So he could not form any new memories. The doctor that worked with him for 50 years, every day she had to introduce herself to him because he just wouldn't remember that he already met her. 50 years. I know, isn't that crazy? So severe anterograde amnesia. He also, because if you remember going back to the declarative versus non-declarative, his medial temporal cortex, that is for your declarative memory. So his declarative memory was impaired. But his non-declarative or his procedural memory was okay. So he could play the piano, but he couldn't remember that he knew how to play the piano, if that makes sense, because his procedural was still okay. So, yeah, really well-known study. The DMNS is, I forget what it stands for. I think it's delayed something. But it's the example he had with the monkeys, and there was new food underneath each shape, and it was, so the monkeys had to figure out like, okay, the food was under the pyramid, and then the next time it was under the block, and they'd be able to figure it out. But then they did lesions. Again, the medial temporal lobe. So again, they took out the declarative memory. And they found that they lost their recognition memory, that they lost the ability to recognize, like, okay, look for the new object, because they lost that declarative ability. Memory. Mm-hmm. And then the last experiment was the Morris water maze. And in this experiment, there was this pool, and just like at home, like if you have a pool and a dog, like, and you put the dog in the pool, they know instantly just go right to the step and get out of the pool. At least with my dog, he did. And so they put these mice in a pool, and they swim around 1 billion times until they accidentally find the step. And then when they put them in the pool again, they go immediately towards the step, so they could remember where the step was. And that uses your hippocampus. That's for spatial memory, because they found that when they did lesions to the hippocampus, that the mice that originally were able to find the step were all lost again. So, lesion to hippocampus, loss, spatial memory. So those were my take-home messages for chapter 24. I went through the list. I did skip a couple of stuff. If you guys want me to, I can go back and go over those, the ones I skipped. But if you guys have questions, we did make it through the material in time, so that was really good. Sometimes, you know, you're rushed and you realize, like, oh my gosh, I only have 15 minutes and I have to do 3 more chapters, you know. But what do you guys want to do? Do you have any questions with studying, or do you want me to go over the rest of the stuff I skipped? Let's go over that. Okay. So let me erase this. And before I forget, I'm going to write the WordPress website where you can find all this good information. Everyone can read that, right? Okay. I also on there have posted old study union videos, so if this one, you can go back and check some other videos out with the same subject, and that might help. They're right on there, too. Okay. Channel proteins and carrier proteins. So these are both transmembrane proteins. So they're getting molecules across from the membrane. Channel proteins kind of look like the KDR, where they're just that. So they can either be open or closed. Carrier proteins they look more like this. And then so they grab something on one side, grab it, and then do a protein conformation and then turn it around and dump it out. Carrier proteins are better for larger molecules, and they're slower than a channel. Proteins, they're better for smaller molecules, and they're faster. You have open and closed channel proteins, and you also have gated and not gated. So under gated, you have an example of 3 types of channel proteins. You have voltage gated, which we talked about with action potentials. You have mechanically gated, which we've talked about with hearing, with those stereocilia. Those gates were mechanically gated. The movement of the stereocilia opened the gated channel. And then you have ligand gated. And ligand gated is basically, it needs a neurotransmitter to bind. So once the binding of a neurotransmitter will allow ions to come in. Okie dokie. Saltatory conduction is basically just a name for how action potentials work. So if this is the axon and then this is the myelin, the ions can't access the membrane through the myelin. They can only access it during the nodes of Ranvier. So the ions are going to come in. So they're going to flush in, and you're going to have your action potential, and then your action potential kind of shoots or jumps through the myelin, and then you have more ions come in, and then it jumps. So that's why it's called saltatory, because saltatory means jumping. So it jumps from nodes of Ranvier to nodes of Ranvier. Does that make sense? Some other neuroscientists. We have Sherrington. He discovered that he was the one who coined the term synapse. So I always remembered SS. Louis, Otto Louis, he was the one who found the first neurotransmitter, which was acetylcholine. And then Frischpan and Potter, they were the ones who found electrical synapse, which we're going to talk about the difference between electrical synapse and chemical synapse in a second. And I always remember that because of Harry Potter. He had a lightning bolt on his forehead, electrical. It kind of goes hand in hand. Ooh, falling all over the place. Okay. So electrical synapse versus chemical synapse. We kind of already talked about chemical synapse with the neuromuscular junction and everything. Electrical synapses, they use gap junctions, where chemical synapses use the synaptic cleft. Electrical are faster, and they're more rare. Also, electrical has a channel protein called connexon, which is made up of 6 subunits called connexins. So that's just a little fact. Chemical uses neurotransmitters. More common, a little bit slower. And they both are used to communicate. Recycling. There's a couple different ways that neurotransmitters can be recycled from the synaptic cleft because not all of them is going to find its receptor. So the first one is diffusion. It's going to just diffuse away. Second one is reuptake. The presynaptic cell is going to reuptake it. And then the third would be enzyme degradation. And that's how they're recycled. And that kind of goes hand in hand with serotonin with an SSRI, which is a type of antidepressants, and it stands for serotonin selective reuptake inhibitor. So you're preventing reuptake, so you're preventing recycling of neurotransmitters, which is going to cause more neurotransmitters to be in the synaptic cleft, which means a better chance of a neurotransmitter binding to a receptor. Do I need to write that down? Or does
Can you say it one more time?
Yeah, so since this is a reuptake inhibitor, you're preventing reuptake, which is a type of recycling. So that way you're maximizing the amount of neurotransmitters in the synaptic cleft. Because if they get recycled, then they leave. But if you prevent them from being recycled, there's a better chance that they're going to bind to a receptor. Does that make sense? And the more serotonin binding, the happier we are. Oh, I totally forgot about glutamate receptors. So let's talk about that. So glutamate has 3 receptors. It has AMPA, NMDA, DA, and kainate. We don't really know much about kainate, but I would definitely know that it's just a receptor. And this is going to go hand in hand with chapter 24, because I noticed he put that on 24, long-term potentiation. Long-term potentiation is just basically how we can form long-term memories. And how we do that is by building NMDA receptors.
By doing what?
By building NMDA receptors. So here's AMPA, and here's NMDA. And these both are ligand gated, so they need glutamate to bind, and then ions can come in. So I don't know if it's 1 or 2, but I don't think he specifies in his slides. But 1 or 2, glutamate bind, and then you're going to have sodium influx. And we know sodium influx is going to depolarize the membrane, because over here at NMDA we have a magnesium block. So even if glutamate binds, the magnesium block can't come off. So the depolarization of AMPA causes that magnesium block to come off, and then NMDA can then depolarize and cause. And he had a picture in the slides of the cycle of how you build NMDA. I don't know if that's necessarily important or not, but at least you can look it over. But you need AMPA to work to make NMDA to work. So this is kind of like his wingman, so to say. Okay, and then there's one last thing on my list. Which is the pre and post central gyrus. So if this was the brain, we have our central sulcus. And the central sulcus is about like right here, and it divides our frontal lobe and our parietal lobe. And then the area before the central sulcus is called the precentral gyrus. And the function of that is, because since we're in the frontal lobe, is motor. And then behind it, right immediately behind the central sulcus, is the postcentral gyrus. Which function is sensory. Also, he had in his slides like a picture of like this. And here would be like the central sulcus. It was like 1B, 1A, 2, and then other. And just know that this part is your S1, which is your primary sensory or primary somatic sensory cortex. And that's located like right here. So it might just be like a question of like which Bowman numbers are located there.
So what are
1A, 1B, and 2, I believe. Let me double check my notes because I know I drew them out. Oh, it's 3A, 3B, 1, and 2. So good thing I checked that. Three A, 3B, 1, and 2. So other than that, do you guys have any other last minute questions? Sorry that the doc cam wasn't working and we had to go based off of the whiteboard, but other than that, I hope you guys do good on your exam. Good luck, you guys got this. Check out the website. I have so many materials, like it's just a laundry list of just like handouts and packets and videos and everything. So check it out and it'll definitely help you feel more confident for your exam. So, alrighty, clean this up.